This be all the glow things?
Yep, tomorrow.
Can I attend?
Yeah. Yeah, we should have enough supplies for another person going on.
Yeah, that's why I'm asking, because I think there's some of these weird molecules where like you expect that when you move down into like all those structures.
We forgot about it. We actually had to move it. Because it snowed before, so it was because it was a month ago. Or
Yeah, well, I think like when you get to snow.
and it's just iced, and then yeah, it was mostly a problem because the instructor was in New Jersey and needed to go to New Jersey after class, so we didn't want her to expert.
Yeah.
Which could be
Yeah.
fine tuning.
What do you use?
I build that research.
Oh.
I build that research. How are you?
Hello. How are you doing? Yes, we are. You might. So we are in the front parking lot. Moving the gated area. I think I see you. Yeah, it's just good to be in the middle. Yep. We're with the light on over here. You're also.
Yeah, I'm surprised you
Okay, you should put them up.
Yeah.
Hey,
Hey.
I know, you have another event going on. So.
Yeah.
A little bit.
Kind of blown out.
Oh, nice.
I should probably
You've been enjoying your wedding?
busy? Running away, that's a good thing.
Yeah.
Slowly getting everybody ready. Going down the street and we're like, oh,
Yeah, it's south of here. It's a BD.
I do have anybody that's
It's right on the Main Street.
where do you live?
Yeah. You don't have to take me all the way. Just throw me out on campus. Take the shuttle.
Where might you at? Well, so this is like, I don't know if it's like, hopefully I understand your thing. And like, it's part of my never dating, like, I'm not getting there. You should not take this car.
So what did you
I'm driving to the
That's what I learned when I was in middle school.
Oh, okay.
I got to the point where I was,
if you want to
Well,
yeah.
it was all about. So we're gonna try to
Is that okay? Or if you're not, I'm just like very nervous for this.
It's a cellulose.
Okay.
Slur.
That,
And
no.
there's a
Okay. But hopefully
Freeze
Oh my gosh.
use that. Takes me a while to get more interesting. Like, oh, yeah, starting growing up. Haven't really got to see them yet. Hopefully, like 5 or 6.
If it's a penny band.
Just very
Initially, companies.
Right, that's exactly, yeah, I mean, that's exactly what you have to do because, like, almost to the point of, you might even be better off to have some people projects. My side, it's just like, kind of like, right, it's more like. It's a hard thing to do, right? So it's something that can just make sense in the future.
Like anything else, we
Yeah, same thing. Talk to Jen.
Yeah.
Like she would, I'm sure.
We were talking about this here. Medical jargon, that's what jargon, whatever words you toss out. We had a I'm gonna see if I can be Hey, hi. So we're asking
I occasionally think
Endangered species.
I actually found myself in very like
I don't think you can do it.
You have to eat your email to yourself or different, but you look like
We're becoming a party. this here.
I did.
Yeah, I was like, you got here. I just
Outside.
I don't know what she is. She lives in
Almost like.
Yeah.
Yeah, that's also.
Really? Yeah. I think she'll have like Ireland. I know she's figured out that.
I know people who work in that area.
Oh, really?
You know, so. So,
I am. Yeah, the suburbs.
like 8 hours.
Yeah.
We have to finish these races and then I have to condense it down. We could actually do it. These are I'm running off like one source and then a source that's like that. And I'm like, well, it's actually fairly hard to get this.
I was like,
Yeah,
Bourbon.
No. Governments. The way that's they started as my Facebook account was just like, here's more money. Do and
I'm like, what am I
it's like, all right, here's all the things that say these civilians who study Finland.
Essentially, like, eventually,
Got around to it.
some number of spots that are supposedly easy.
We're gonna start in a few more minutes. But if anybody needs water, beer, coffee, feel free to go in the back and get some. We have 6 scientists here to talk today, and we'll get started in a few minutes. And if anybody's new to BUGSS, I've already gotten some people. I'd love it if you filled out this really quick form, so like a little mini questionnaire, because it helps us with our data. Everybody loves data, so thank you. Thank you. Let's pick the other one back here. Hey. We do, yeah. Oh, right back to the.
You're on the history page.
Thank you.
If you were to like grab anyone on 7, you would be completely normal. Like you're not one of those like, oh yeah, so they just lounge. Yeah. Switch labs. You take your classes and then oh yeah. There's nothing. I haven't interacted with him. He developed that new biostats course that replaced Doug Frost. Yeah. So I only saw him for that interaction. I took no, that's like
You don't understand what you're doing. You don't understand how. They told me all kinds of things.
oh, you didn't use I can now make yeah, not in touch. I would
Yeah, so we have kind of work on some other organizations.
Yeah.
And then class.
Would she have to be monitored?
Yeah.
What does she do? By membership?
How about you?
We've been doing it.
Do it now.
We've been doing
I didn't
it.
Oh, you got your son. Placebo.
I don't know anything about that.
Yeah.
Not tonight.
All right, I think we're ready to begin. So the first thing I want to say is it's Friday night and we're going to spend the next 2 hours talking about science, and that is awesome. So good for you. High fives all around. So welcome to BUGSS. So for people that haven't been here before, I'm Sarah Lon, and I'm a very much lucky person that I get to be the lab and program manager of this remarkable and quirky community science space. And that's exactly what we are. We're a community science space, and we kind of have I want to make sure that I say this in the way that I want to say it. So we are into many different things, and the overarching goal really is to provide community with science knowledge, education, and resources, and a fully functional lab. We believe, everybody should be able to learn and practice science. So this is our space, right? So you might be wondering how, if you are really feeling like you want to volunteer or be a part of this movement of, getting science out into the public, you can volunteer here. And just one of the ways you can do is mentors. We have community projects. You can come and mentor the projects. They're pretty fun. An example is we're going to grow some mushrooms and make them into molds of biodegradable or usable, really, pots, flower pots. Another one would be where we have a yeast sensor that's going to detect estrogen in the environment. We also have an iGEM team of high school students, and they're in here this summer. And this is not just talking to kids about the organelles of the cell. These are smart kids. They're doing synthetic biology in the lab, actually making plastic-eating bacteria. So it's a lot of fun if you want to do that. And then finally, of course, if you're all academics here likely, or most of you are, and if not, then you're in a full-time job. You might not have the time to dedicate. And if you don't, you can support us with a supporting membership. It's pretty kind of like WYPR, the idea that you're supporting the group. But in saying all that, I want to say thank you so much for coming here, and thank you so much for these speakers, because it is more important now than ever that we have science out in the public. And I thank you so much for being brave enough to come in here, and for Project Bridge. So Project Bridge we're like a co-host here. So Richard is going to come up and talk about Project Bridge a little bit.
Yeah, so I'm Richard. I'm part of Project Bridge. So basically, I'm a graduate student at Johns Hopkins, and Project Bridge is a student-run organization that's trying to bridge science with the public. So we hold science cafes where we bring researchers to give informal shop talks in a nice little cafe or bar somewhere. We have a booth at the farmers market in Waverly to give hands-on demonstrations to little kids. And we recently, or I guess a few months ago, had our first brain festival where we had school rented out. We had many different labs bring in their equipment to play with. So you see kids playing with prosthetic limbs and things like that, or hearing talks about sleep, or doing an escape the brain room kind of puzzle thing. But I mean, basically, I agree with Sarah. We are trying to get science out into the community because too long, many scientists are content to just be in the lab to do research, which is important. But I think now more than ever, it's important to make it clear why what we're doing is good and necessary. So So Project Bridge, like BUGSS, wants to make science more appreciable and understandable to the general public. So this is really great to have our third science lab here with BUGSS. I don't know if this was mentioned before, but there are prizes for tonight's speakers for both the most creative talks and the best communicated talks. And these are in the forms of Amazon gift cards, which you can use to buy things, I'm told. But yeah, I think that's it for me.
Yeah. And
I'll be live tweeting, so if I'm on my phone, I'm not distracted. I'm actually.
What's your
trying
Twitter
to-
handle?
Oh, it's just my personal one right now. It's Richard Sema at Richard Sema, and I'll be tying it to the Project Bridge account, which is Project Bridge on Twitter. So.
And if anybody else tweets, I've been using the hashtag SciSlam, SCI Slam, just for going out there. I actually contacted a couple of podcasts that were interested, so maybe people will actually be listening to your tweets, so reading them. Yeah, okay, just to also clarify, those little pieces of paper that you have and pens, what we want is constructive feedback for our speakers. You know, this public speaking is hard. Also, boiling down your research, your passion for likely the last 5 years or so is really difficult, right? So be very appreciative of that. But be, constructive feedback. For instance, I talk way too fast. So people always say, hey, slow it down a little bit. So things like that on the cards were really useful for them. All right, so with that, are you ready? All right, cool, all right. I'll let you know if we had, last time, we had a very diverse group of people come, and when all the titles were coming in for this round, I was like, wow, everything's about the brain. This is really interesting, except for a few interesting oddballs, right? But this is gonna be really fun. So right now, we have Sarah. She's going to talk about sleep, neuroinflammation, and the developing brain, which I have a feeling is going to scare me because I have a 4-month-old and I don't sleep at all.
I said this at a conference, I got that conference. So I'm going to try not to scare you too much. Okay. All right, hi. So I'm at Johns Hopkins. I do sleep research and I'm kind of a pediatric population. We'll get more into that. So in your first couple years of life, so in the green squares where we're going to concentrate the first 2 years of life, you spend the majority of your time asleep. So you spend between 12 and 16 hours of sleep a day, which is a lot. And within that, you spend an inordinate amount of time in REM sleep or rapid eye movement sleep. That's what you think of when you're dreaming. So you spend between 30 and 50% of your time when you're a baby in REM sleep. And what's not shown here is you also spend a very high proportion of your time in stage 3 sleep, which is your deep restorative sleep, where there's a lot of delta wave activity, which is thought to be pretty important. Also during this time, during your first year of life, your brain is developing. We're still learning a lot of the intricacies of that, but we do know that sensory pathways like vision and hearing, your language pathways are developing. You have higher cognitive function pathways that are beginning to develop. So there's this idea that during this first year of life, there's this critical window that's happening that may be tied to sleep because you do spend this large chunk of your day asleep while these pathways are developing. In children, there are sleep disorders. It's very small, sorry. So about 40% of children, parents report some sort of sleep disturbance in their children, whether it's about 30% will say, hey, my kid's not staying asleep, they're having trouble falling asleep, they're having night terrors. 6% of parents report their child is always snoring, and 4% actually are diagnosed with obstructive sleep apnea. That's when the airway actually closes, they stop breathing, and then they wake up, just fragmenting their sleep. There have been some studies showing that if you fragment the sleep of children in early life, so parents report their kids aren't sleeping well, if you look at them a few years down the road, they're not learning language as well, they have problems with attention, performance on some executive function tasks like making choices. These are all impaired later in life. However, the function behind that is not well known or known at all. We also know there's a higher proportion of sleep disturbance in children with neurodevelopmental disorders like autism, ADHD, cerebral palsy, and Rett syndrome. So is this the chicken or the egg? Do sleep disturbances result in neurodevelopmental disorders, or do these neurodevelopmental problems result in poor sleep? It's kind of hard to ask this question in human children because it's unethical to purposely wake up babies for a lot of reasons. Probably also unethical to do that to parents. So what happens if we artificially fragment sleep? So in our lab, we looked at the literature, and so in adults, if you look at this, you see that there's microglial activation. So the immune cells in the brain are becoming activated and looking for things to get rid of. There's an increase in inflammatory proteins called cytokines. And you can also see impaired performance on vigilance and attention tasks and learning and memory impairment. Anybody here who hasn't had a good night's sleep can attest to that. So what does this do to the developing brain? So we looked at this in rabbits. We fragmented their sleep for 3 days at day 3 of life. And this is 7 days after we fragmented their sleep. They've been put back with mom, haven't done anything to them. On the top, we have our fragmented animals. The green is microglia, and the blue are cell bodies. And then the cortex and hippocampus, so 2 areas important for learning, have an increase in this inflammatory cell. So there is this evidence of a sort of chronic inflammation occurring. So this is 7 days after fragmentation. So we went a little bit further out and looked at a behavioral task called the novel object task. You put the animal into an arena, and there are 2 objects that are identical. They're in there for 5 minutes, and then they're taken out for an hour and they're placed back in, same arena. One object is the same, one is novel. And we measure the amount of time spent with the novel object. In theory, they want to spend more time with the novel object because it's new. So we actually, here's a picture of one of our rabbits looking at our novel objects, also looking at me. And so what we found, so this is about 3 weeks after their sleep has been fragmented. Nothing else has been done to them. We see a reduction in the amount of time spent with the novel object. So they're impaired in figuring out that, hey, this is a new object. I've never seen this before. I want to look at it. So it's actually a pretty significant amount of time that is spent. So there's some sort of learning and memory impairment going on, which we're exploring a little bit further now. So in summary, in our healthy rabbits, we disrupt their sleep at a very young age. We have neuroinflammation that's characterized by this increase in microglial activation. We see an increase in inflammatory proteins. We actually see that this persists for up to 2 months. So in humans, it's a couple of years. We see changes in learning and memory performance. We also see chronic changes in inflammatory cytokines in key areas for learning and memory, like the hippocampus and cortex. And together, we think that this indicates that it's really important for kids to get sleep early on, and healthy sleep and consolidated sleep. And with that, I'll say thank you to my lab, to my funding, and for all of you for listening. Excellent work.
Is that your rabbit, sir? High five?
No, that's a Google rabbit.
Why do you use rabbits?
Rabbits actually have a very similar brain maturation rate compared to humans.
Okay.
So unlike rodents, which take about 3 weeks to mature their brains after they're born, rabbits right out the gate are there. Also, we have to separate them from mom for 3 days, and it's a lot easier to hold a rabbit and feed it by hand than a mouse.
All right, I'm so sorry. So actually, we're going to hold questions till the end, so that way, so we're going to have a, but you're the executive director of BUGSS, so you're allowed. Just one question. All right, so we'll have a kind of a Q&A after, we kind of have a break after everybody talks. So next up is Paige, and Paige is going to talk about pain in the brain.
If I can find it. I minimized. Oh, this one? Yeah. Yeah, there we are. All right. So where is it? It's not showing up here.
Is it the split screen thing where you have to drag it to the opposite side of it?
Oops, not that way. Oh, that's very good at that. There you go. You grabbed it. But it's that way. There you go.
Hey, here we are. And now I can.
Don't worry, I haven't started the timer.
Oh, there's a timer. That's okay. Alright, hi, I'm Paige Sedlak. I'm a fourth year PhD student in the program in neuroscience at University of Maryland, Baltimore. And today I'm talking about my research with pain in the brain, but I'm really going to give you a more general overview of what we think is happening because there's still a lot of questions we don't have answers and ongoing study. So we usually think about our bodies sensing the world around us as just kind of a one-way street. Like your nerves pick up on some stimulus in the environment, that information travels to the spinal cord and then up to the brain, and the brain processes that information. But really, it's a lot more complicated than that. With pain signaling, there's another pathway that acts in reverse. The higher order areas up in our brain are taking into account all the environmental and emotional factors happening around us at the time, and just weighing this balance of how important it is that you feel pain at that moment. So if you're still in danger and you need to survive, that pain may not be felt yet. However, if you're safe, you're not distracted, feeling pain is important. It teaches you to avoid things that will injure you, and it also encourages you to rest that injured site and let it heal. Yeah, to further complicate the pain transmission to and from the brain, there's at least two distinct pathways going from the nerves to the brain. So the sensory pathway is the classic. It tells you what hurts and what kind of pain that is, like burning, stinging, that kind of information. And there's another pathway that I personally think is more interesting because less is known about it. It's the affective or emotional pain pathway. It tells us information about the feeling of pain as a negative emotion, how unpleasant it is. And I think affective pain may hold the key to some good chronic pain therapies because the sensory pain pathway has been well studied, much more than this affective pain pathway, and still has not produced great treatments. Things like opioids, you can't use them in the long term, and they don't work for everybody, and they have all these other super detrimental consequences of use. So while all these pathways, the ascending sensory and affective pathways and the descending modulatory pathway are in play during regular pain, they're also present during chronic pain. Chronic pain is a fascinating neuroscientific state. The pain that you get from this injury somehow changes your brain. So the injury is healed, but your brain is still responding like there's pain in that site. And not only does it think there's pain in that site, but that pain can spread. So we know this because if we cause a nerve injury here on this nerve branch that governs sensitivity of the cheek and whisker pad in a rat, after a few days, those rats are sensitive in the other parts of the face, showing that like as far back as the trigeminal ganglion where these nerves converge, that part has changed already. And then a few days later or a few weeks later, the other side of the face or remote parts of the body may be hypersensitive. That tells us that the brain itself is changing, that this is no longer a problem in the periphery of the body. It's now your brain has decided that you are in pain and isn't processing this normally. So my lab mates and I are looking at brain areas important for both affective pain and descending pain modulation, and we've come to these two brain regions, the amygdala and the parabrachial nucleus. The amygdala has long been studied as a brain area responsible for fear and threat learning and anxiety, so it makes a lot of sense that it could be processing some emotional aspects of pain. The amygdala normally applies a brake on the activity of the parabrachial nucleus, but that seems to stop working in our chronic pain models. So the parabrachial nucleus cells just fire for minutes and minutes and minutes after a stimulus is presented to the pain-sensitive site. So you can see an example of this at the bottom. So acetone or like nail polish remover, if you get it on your skin, it'll feel cold for a second. And normal rats don't care about that. They sniff it, they're like, my paw smells weird. But they don't respond with any pain behaviors. So, however, this rat had a nerve injury. So I put a drop of acetone on its foot here, and this is a parabrachial cell firing. And this is the time scale. So you see it fires, and it fires fast for minutes. That's just kind of this like runaway train of the brain overreacting to what shouldn't even be a painful stimulus. So we think that this activity is a sign of the overreaction of the brain in chronic pain, and a sign that this descending pathway is losing its balance. We still have obviously this is opening a lot of questions, so the questions we're currently trying to answer are why is the amygdala losing its control over the parabrachial nucleus? We don't know this yet, we're currently studying it, and also, is this a good target for chronic pain therapies? So chronic pain as a disorder comes with a lot of other things, mood disorders and things that could be like depression or anxiety that could be using these same like now dysfunctional emotional pathways. So this is a current target of research in our lab, and that's all I've got for you today, so thank you. And I gave you some more dog pictures. That's Ruby, yeah.
Got little dog pictures. Okay, so next up we have Samson, and he's actually we're gonna mix it up a little bit right now. He's going to talk about crowdfunding, which of course is pretty important.
I wasn't told I could bring a thing,
Oh
PowerPoint presentation, so I'm gonna do it old school and write two things down here. Ready? RPHC. We'll talk about this. So I'm not a scientist. I was introduced as one, so I'm going to claim it as an alternative fact. I'm going to update my resume. So
wait. This is not on the timer. What did you study?
Oh, okay. I went to Florida State University, the greatest university in the world. I studied anthropology in undergrad.
There you go.
It's a science.
Okay. Are you an anthropologist?
No, but I love the field.
It's an awesome field. That's what Florida State means, yes. We don't actually have academics here, we have a football team. But from anthropology, I went to study medical anthropology. I went to become an epidemiologist because I wanted to know what medical anthropology was. And from there, I went to finance and have done horrible things for evil empires in finance, which is where I learned a little bit about what the fuck is crowdfunding. And so there's 4 types of crowdfunding along the clock now. There's donation, where you're literally giving away money. You don't get anything back. And then there's rewards, where you give me some money, I'll give you a reward. Gum or something, whatever I happen to be giving away. Then there's some other more exotic types of crowdfunding called debt, which is known as peer-to-peer lending. Everybody in here reaches deep into their pockets and gives me $100 at a slightly different interest rate. Sarah gives it to me at 3%. He gives it to John, gives it to me at 12%. And so the blended rate, it comes out to about 9.5% typically. These are data from Europe. The other type of crowdfunding, this type of crowdfunding I do quite a bit of, it's called equity crowdfunding. Works just like Shark Tank. That's it. It's really not that much complicated. So crowdfunding, it might seem like really nuanced and complicated, but it actually is pretty old. If you've ever been to church and they pass that plate, that's crowdfunding. The only difference is how we do it. We use one of these little devices. We have the history of the world, and I mostly use it to watch YouTube videos. But that's how you crowdfund. And crowdfunding means you get people who are interested in a thing, and you go out and you compel them, and you tell them a good story. It's like, this is why you should give me money, or invest in money, or lend me money for this thing. So the reason I'm here today is because I'm working with Tiffany Gray, who's not here at the moment. She had to go to Los Angeles. When I'm not being an evil person in the world of finance, I'm on the DC Department of Health Institutional Review Board. We read a bunch of grants and stuff. And I was sitting there and I was thinking, how much work goes into this junk, this stuff, right? And so I asked the big brains, I was like, big brains, when you send us this stuff, how much actual money goes into this? So they went back and forth and they did it way too academic-y. It's about $17,000. If you submit a grant, 95% of grants get rejected, and that $17,000 you might as well burn. And I was like, crowdfunding. For the brain research, the pain research, if you do the work and you put it out to the academics, would they be interested in funding your work? I don't know. But that's what I'm working with Tiffany from, she's a doctor of public health at George Washington University in DC. That's what we're working on figuring out. How do we take the academic research that everyone already does, 95% of which gets thrown away, and figure out a way to put it to their peers to say, instead of voting whether or not you like it, vote whether or not you want to have an equity in this idea, or if you want to lend them money, or if you actually want to donate. So I like the equity in the lending aspect of it, because that's a business. I'm not asking you for money, I'm asking you for an investment. Because there's lots of ideas that they might take a while to gain fruition and work, but if you've done the work and there's a bureaucrat who rejects you because they don't have that vision or that foresight, they're not looking over the horizon, I say give it to your peers and let them vote with their wallets. So that's what we're working on. It's like a side project to my actual job, and sometimes they bother me, like, Samson, why aren't you working? So that's crowdfunding in a nutshell. Super excited about hearing your questions at the end of this.
I think that's a good idea. All right, so now we've got Brian. He's going to talk about another sleep talk, which is kind of interesting. So this is sleep modulation to influence human performance. So I'm very curious.
Okay. Well, thanks everyone for having me. So my name is Brian. I'm a PhD candidate at University of Maryland School of Medicine, and I teach in the physical therapy department. And I have no disclosures. Okay, so I'm just gonna, break it down very simple since I have 5 minutes here. So what I'm interested in is rehabilitation. So when someone has a stroke or a brain injury, a lot of times people say, well, you need to relearn how to walk again, or you need to relearn how to brush your teeth and things like that. And so what I'm looking into is the role of sleep modulation in learning. In this case, this frame I'll be showing you is just of healthy young adults, but the idea is that you can scale it to individuals who've had a brain injury. Kind of piggybacking on the previous sleep talk, if we look into how the brain maturates after birth, there's actually very interestingly a similar type of maturation which occurs after the brain injury or after the stroke, where it seems like there's this going back to the beginning and having to mature through sleep the first few months afterwards. But anyway, so when you're learning a task, whether it's a cognitive task or in this case a motor task, there's the initial phase of encoding or studying or practicing, whatever you want to call it. And then there's this few hours where there are very specific molecular processes going on, which I won't get into here, but those are known as consolidation. But a lot of what people are looking into now is what the individual is doing during that 8 or so hour period can influence their retention of that information. So during the daytime on the left here, whether it's a motor task, if you're playing golf, or a cognitive task, if you go read a new book, that's going to actually interfere with that memory you encoded to begin with. Whereas oppositely, at night during sleep, there I just have some sample EEG traces of the brain, but the brain will actually spontaneously refire the same neural networks and areas that were fired during actually throwing the ball. So it's almost like you're reinforcing it. So I have the little picture there, been thinking about it. And then of course you have retrieval, so throwing the ball again. But what I'm interested in is, can we use an intervention which we call targeted memory reactivation to enhance this consolidation process, in this example, to help these young people learn to throw this ball better with their non-dominant arm. So it's a little bit more challenging. So the idea is kind of going back to what I just said. So we had people use their left hand to throw balls at different targets, and then use an auditory stimulus to pair with the throwing. So it's kind of like the old Pavlov experiment where you ring the bell and give the dog some food, and then they associate the two. So the idea though is if we ring the bell later during that consolidation phase, will it trigger the brain like it was going through sleep again? With the hypothesis that if we do it during the daytime, it'll have a detrimental effect because of what I was just talking about. There's other things you're doing, so it's going to impair consolidation, whereas at night, we hypothesize that it should improve consolidation. So this is the first cartoon here, just kind of showing what people were doing. They were at the University of Maryland Hospital with me overnight, and as they were asleep, I was playing these cues for them. And what we found is, so there were 4 different groups here. So there's wake, people who stayed awake during their normal daytime hours, or they slept during their normal nighttime hours. And then they either did get the targeted memory reactivation or did not. So that creates 4 groups for us. And so we see that if you look at the change score of their accuracy of throwing the ball overnight, and then here it's normalized to their baseline performance, what we see is that the people who slept and got the auditory cues from me are the only people who improved overnight. And then actually a week later, we still see quite a bit of difference between those 4 different groups. Although here it's not statistically significant because the sample was probably too small. So then it seems like playing these cues had different effects based on the sleep or wake state of individuals. So then the next question is, can we use this as an adjunct to physical rehabilitation after someone's had a stroke or brain injury so that, again, they can learn how to walk faster or have a higher ceiling that they can reach? And that's it.
Whatever.
So next we have Elizabeth, and she's going to talk about neuroinflammation and nanoparticles.
What is the particular topic?
Where is the mouse? It's on the
Oh,
screen.
It's on the, you gotta bring it back to the left. The other way.
So it's on that side. Come to us. I swear I have PhD.
Am I hitting escape?
There, I found it. Nice. Okay. Oh, gotcha. All right, we can see it. Great. So I'm Liz. So I'm going to talk to you about neuroinflammation and nanoparticles. So Sarah talked a little bit earlier about neuroinflammation and sleep. And it's something that, I work with Sarah, but we have a larger lab group that has a lot of diverse interests, and one of them is using nanoparticles. So very briefly, inflammation, when we talk about it, and by definition, is a protective response in which we are trying to destroy or get rid of either injurious agents or the damaged tissue. So we have inflammation that occurs systemically, but we also have inflammation in the brain, which we refer to as neuroinflammation. And it seems to be, and so here's my awesome Google image of a macrophage swallowing up a virus. So we have diseases and disorders that are characterized by neuroinflammation, and they run the gamut from acute injuries like traumatic brain injury to progressive neurodegenerative diseases like Alzheimer's disease, and down to developmental conditions, including cerebral palsy, which I work in, as well as autism. And some are starting to think that mood disorders have some component of neuroinflammation like depression and bipolar disorder. So you can think of neuroinflammation in the process as, instead of the good, the bad, the ugly, but the bad, the good, and the ugly. So the bad being there are toxins or pathogens that can cause damage to neurons in the brain, or in general just damage and disrupt everything that's going on. So you have the good being the microglia. So these are your immune cells in the brain. They come and they rescue your neurons. They come and they will clear out
all the pathogens, and they'll remove the sick and dying cells so that you can, go on as business as usual. However, this process can get hijacked, and then you have what results in this perpetuating cycle of where microglia stay chronically activated and continue to secrete their neurotoxic factors that help eliminate sick cells, but instead of eliminating sick cells, you are eliminating healthy cells. So it continues this process when you really don't need it anymore, and that's one of the things that's still under debate and under study in regards to Alzheimer's and Parkinson's. But at this point, we know that neuroinflammation is an important component, but we also know that we have a lot of trouble treating neuroinflammation in the brain. We have anti-inflammatory drugs and antioxidants that we use, and they work great systemically, but the problem is getting them to the brain. A lot of times these drugs get metabolized or used prior to making it to the target cells in the brain, so they don't get to the microglia that really need them. And if you give a dose large enough that gets the drug to the brain, you end up with offside effects or side effects that are really detrimental. We know that large doses of NSAIDs end up with renal failure, so we can do better. And so one of the ways we want to try and do that is by using nanoparticles. In our lab, we use dendrimer nanoparticles, which are these hyperbranched structures that are built with the same building blocks, one on top of the other, to create this ring-like structure that looks kind of like you're looking at the top view of a tree. And we can attach things to the outside layer of the dendrimer. And when I talk about dendrimer nanoparticles, I'm talking about something that's 4 to 10 nanometers big. And so when you're thinking about something 4 to 10 nanometers big, it is in relation to the size of a soccer ball that the soccer ball is to the earth. So that's how small we're talking. If you can wrap your brain around it, it's very small, and it allows it to penetrate the brain very, very well. We also like dendrimers because they're not very toxic, and you can easily make them into solutions or even or oral dosages as well. So very easy to give to people, and currently right now animals. But the other thing we really like about them is that we can protect these drugs from being metabolized or used before reaching target cells. So the way we attach them to the nanoparticle, to the dendrimer, can keep them from being used, and we can get them to microglia. And we know that we can preferentially target microglia because they're already angry and all they want to do is eat up things that shouldn't be there, and dendrimers shouldn't be in the brain. So we have this advantage already. So what you see here is, we've got, it's a little hard to see, but you have your nuclei in blue, microglia in green, and your dendrimer, which we've tagged with a red tag here. And we have cerebral palsy rabbits that we studied, and you can see that the dendrimer is taken up into these sick microglia, so you see this overlap yellow, but we don't see the microglia in these, or we see the microglia in healthy rabbits, but we don't see the dendrimer being taken up. So the other thing is that we can preferentially target microglia, but also activate the microglia. So it's not going to resting ones, which is great because it means we're getting the nanoparticle to the site of injury where we need it to go. So in summary, neuroinflammation can lead to healthy cell death, which we don't want. And anti-inflammatories are ineffective at treating neuroinflammation generally. But we can use dendrimer nanoparticles to improve delivery of anti-inflammatory drugs to the brain, and specifically microglia. So,
Very nice.
So we all work in a very collaborative environment. So between both of my mentors, between Johns Hopkins University School of Medicine, Penny Krieger, and Wilmer Eye Institute for Nanomedicine. So we're a big group, and we have a lot of things going on. So this is just the tip of the iceberg, but it's a lot of fun.
Nice.
Great.
Back to you.
Ruby is the mascot of the night. Yep. So next we have Amanda, and actually,
Cover up.
yeah.
My talk.